Okay, so we said that enzymes are a certain classification of the function of certain proteins. Not all enzymes are proteins, but most enzymes are. About 95% of the enzymes that we know of are protein in nature. Some RNA molecules can also act as enzymes. We'll see that when we look more at the ribosome structure. But essentially, enzymes are going to be components of the cell that speed up chemical reactions. So it's important to note that enzymes will do this without being consumed in the process. So enzymes can really speed up reactions. They can increase them by 10,000 to 1,000,000 times. So these are very, very important because they make reactions happen within the body that wouldn't happen for very, very, very long times under normal conditions. So both Chemistry 104 and Chemistry 105 have spent a lot of time talking about chemical reactions. So reactions can be of two major types. They can be catabolic, where they're actually breaking things down, or they can be anabolic reactions where they're building things up. But essentially, they're either breaking or making chemical bonds. That's what enzymes do. They're going to cause chemical reactions to occur. So they usually do this by forming an enzyme substrate complex with the molecules that they're going to help change, either build or break down. So the substrate tends to bind to the enzyme at the active site. So the active site is going to be the region of the protein where the substrate actually binds to and gets converted into the product. And then the product will leave the active site of the enzyme. So if we think about this in a diagrammatic sense, we can look at an example here. This is the sucrase enzyme. It's going to break down the disaccharide sucrose. So we saw the sucrose structure last term when we looked at carbohydrates. If we eat sucrose, this is table sugar, remember? So if we eat sugar, the sucrase enzyme inside our body is going to break that down into a molecule of glucose and a molecule of fructose. So there's the glucose and the fructose. So when the sucrose comes in, it's going to actually bind in the active site of the enzyme. So this blue area here is denoting the active site that actively binds to the substrate. Once it's bound in the active site, the enzyme makes the breakdown of these two products more likely to happen. So it speeds up that reaction. Once that reaction has occurred, it's going to allow the products of the reaction, the glucose and the fructose, to actually leave the active site of the enzyme. The enzyme then is unchanged in the process. That's a very important feature about the enzymes. They don't get used up during the process. So they can be used over and over and over again within the body. And that's a very cool feature about enzymes because you only need a little bit then to make reactions go forward. And because these enzymes can be active over and over and over again on new substrate molecules. They never get used up in the process. So at the end of this chapter, we want to talk about one of these classes of enzymes then that breaks down proteins into their amino acid substituents, and this occurs usually during the digestion of proteins within our diet or the digestion of old proteins within our cells. And so these happen very, very specifically so that we break down our proteins into the amino acid monomer subunits. So remember, when we built our peptide by forming the amide linkage, and we did that as a dehydration reaction. Right, so we took a carboxylic acid and we took the amine functional group on the next amino acid downstream, and we pulled out that water molecule and we linked our amide linkage here. So protein hydrolysis is going to be the reversal of this process. Hydrolysis means that you have to add water back to the reaction, and in this case it's going to cause the breakdown then of the primary protein sequence. It's going to break down the amide linkages between the two amino acids. It's going to give back the OH to the carboxylic acid and the H to the nitrogen. And so you see the reformation then and release of individual amino acids as the protease is working on the hydrolysis, or the destruction, of the overall protein structure. So in this case, primary protein structure is also going to be destroyed. So we said that protein hydrolysis is going to occur within cells to help break down older proteins that may no longer be functioning correctly. So just like everything else, our proteins will age, and over time they may get damaged by reactive oxygen species or other toxic compounds that we might be exposed to. So our body's constantly having to make sure that it renews and rebuilds new proteins that are correct in their folding pattern so that they maintain their function. So protein hydrolysis can also occur within the cell to help control protein activity, and this is a very useful feature of breakdown, right? So if you don't want a protein to be active, one of the ways to make it inactive is to get rid of it. So after proteins have served their function, many times they'll be destroyed or they'll be broken down so that they no longer function. This gives a cell a way to have an on-off switch for protein activity. And if it needs the protein again, it would require new synthesis of that protein to have that reaction then. Protein hydrolysis also occurs as part of our immune system, and it helps digest foreign proteins that may enter or interact with our normal body cells. So our immune system cells, like the natural killer cells or the monocytes or phagocytes, they go around and they will engulf a whole bacterium and they'll start to digest it and break it down. And so they have to deal with all the proteins that are within those structures. And then, of course, we've already talked about hydrolysis occurring during the digestion of proteins within our small intestine as part of our digestive process. So this is going to free amino acids for the use by the body to build new proteins. So we are incapable of synthesizing several of the amino acids, and we have to get them from our diet. So some of the amino acids are going to be essential and have to come from the diet to actually get them. Otherwise, we would be deficient in those amino acids. So this section is going to end our discussion on Chapter 18, Protein Hydrolysis.